increasing of skeletal muscle. Also in dairy animals, there are major allergens in milk, which you can knock out using CRISPR, which help make milk more acceptable to those who have allergies to it.
Just need to click that to show.
Yeah.
We've noticed that.
Okay. Hopefully that's the last time we have any issues today. And then we also have the gene editing of plants. So plants are really interesting as well because it's a little bit less controversial to edit plants with CRISPR. And people have already been looking at producing safer and more drought-resistant crops using CRISPR-Cas technology. I'm going to show you a cool example of this next. This is the first CRISPR-Cas field test that's going to go ahead in the UK. This is looking at wheat. So wheat is something we all like to eat. We use it in bread, but this is also true of potatoes as well, is that it contains acrylamide. Acrylamide is a carcinogen, which is obviously something you don't want to eat, that accumulates. Basically, the browner your food gets, the tastier it gets. Anything you bake, fry, or toast accumulates acrylamide. And acrylamide is formed with the Maillard reaction, and it's created from asparagine, which is an amino acid. And what researchers have done is they've knocked down the asparagine synthetase gene, so the gene that produces asparagine in wheat, and effectively created a low asparagine wheat, which will then be a low acrylamide bread. So you're able to increase the safety of our products simply by knocking down one gene. And it's now so easy to do that we're able to do that in the UK. And it's going to be the first gene edited wheat, as I said before, to be tested in Europe. And there's a reason why this is a bit easier to do than has previously been the case, because CRISPR-based systems aren't currently classed as GMOs. They don't contain DNA from other organisms. And this difference might make it easier than the use of traditional GMOs, because when you introduce DNA into an organism from another source, so a GMO is generally something you've taken a gene from another animal or another plant or microbe and put it into your target organism. There often isn't very much control over where that goes. You might have effects you weren't aware of. People are also worried about interbreeding between these plants and wild variants and how you might be introducing foreign DNA into our ecosystems. But CRISPR-Cas based systems don't have to add any DNA from a foreign source, because you can simply edit what's already present in the genome. You're not actually adding anything. It's the same organism. What you're really doing, as I said before, is basically speeding up breeding programs that are selecting for traits, which are, of course, are then related to your genome. So you can skip decades using CRISPR-Cas. Of course, it's going to be important to control and carefully monitor the introduction of gene edited produce, so they don't go the same way of GMOs and don't get trapped in regulatory hellholes. So I'm actually not interested myself. I am interested, but I'm not studying any of those things. I'm using CRISPR for different things. CRISPR-Cas has been a fantastic tool to have in the lab. There's a number of different ways we can use it, from, for example, as I said before, knocking out genes. You can knock down genes. You can increase the regulation of genes. You can take out mRNA, and so many things you can do with CRISPR now. But it can also be used to kill bacteria, and that's because bacteria can't repair the breaks that eukaryotes can. So the reason we use CRISPR-Cas in animals, in plants, is because you can actually knock out genes because it repairs those breaks. Bacteria can't do that. And that comes from the fact that CRISPR is originating from these bacteria. I'll come to that later. And this has also led to the development of a couple of different CRISPR derivatives as well. As I said, you can manipulate gene expression with dCas9, and you can target RNA as well. So you can go even closer than DNA. You can look at the molecule, which is the bridge between the genome and what actually makes us. So I'm going to slightly change topic now into what I'm interested in the use of CRISPR. Oh, before I do that, sorry, I'm getting a bit out of sync here. I wanted to mention briefly that CRISPR has been such an incredible technology that it has been awarded the Nobel Prize in 2020. It was awarded to Jennifer Doudna and Emmanuelle Charpentier. It was a Nobel Prize in chemistry, and this is the first time a Nobel Prize has been won by 2 women. So it is already quite a remarkable technology, considering the paper they wrote came out in 2012. It's only 8 years from the introduction of CRISPR to a Nobel Prize. What I want to do is see if we can apply the technology to assist against drug-resistant microorganisms and antimicrobial resistance. So antibiotics are in their own right a revolutionary technology. Since 1928, when Sir Alexander Fleming introduced or discovered penicillin, they have completely revolutionized medicine. The ability to easily and cheaply treat bacterial infections is something that we take for granted now, but then it saved millions and millions of lives. Even just tuberculosis alone killed millions of people without antibiotics. However, most of our antibiotics are derived from other microbes. So penicillin is from the fungus Penicillium, and a lot of our antibiotics come from soil-dwelling bacteria, such as Streptomyces. And because they are, at least our antibiotics, usually natural products or semi-synthetic analogs of natural products, that means that resistance, natural resistance to antibiotics is also present. And our usage is fueling an increase in resistance to them. So antimicrobial resistance, here we are. This is the big scary thing. AMR, or antimicrobial resistance, is effectively the evolution of microbes to survive exposure to what would have previously been effective compounds. For example, if I had a urinary tract infection 60 or 70 years ago, I could say a course of penicillin would go away. Today, penicillin is almost not used at all in Western countries because the resistance is just too widespread. And drug-resistant pathogens are already estimated to cause 700,000 deaths a year, with nearly half of this alone coming from tuberculosis. It's worth mentioning that antimicrobial resistance actually refers to both bacteria, viruses, parasites, and protozoa, but it's really most dangerous at the moment. We're really seeing the worries in bacteria, because there are bacteria in hospitals which are approaching pan-drug resistance. That is, we're soon going to have no treatments at all. If you get sick from certain bacteria in hospital, doctors have just the hope they haven't got any treatments left. And this is why some have claimed that antimicrobial resistance could potentially be the biggest public health challenge of the 21st century. Admittedly, this came before the COVID-19 pandemic. However, as in the next slide I'm going to show you, I think this does still hold weight. So these numbers I'm going to show you come from the O'Neill report, which was published in 2016. The data comes from 2014, and they estimate the impact of AMR by 2050. So if we look on the left, we can see the number of deaths that are caused by AMR. Now, as I said before, 700,000. In 2050, it's expected that 10 million people a year are going to die from antimicrobial resistant infections, which is more than cancer kills today. This isn't just a health problem, though. There will also be a cumulative loss of $100 trillion to world GDP caused by antimicrobial resistance and resistant infections. So we have both a public health and an economic crisis. And it's already here. We already have resistant bacteria in our hospitals. It's now a matter of how worse is it going to be? How much can we actually do to mitigate this threat? And why is it that we have this problem at all? Well, antibiotics, these miracle drugs, are not a particularly attractive thing for pharmaceutical companies to produce. The last antibiotic class that entered clinical use did so about 30 years ago. And this has led to, as I said before, some hospital-acquired infections becoming very difficult to treat. If you look at this graph on the bottom here, you can see the years in which antibiotics have been discovered. For example, penicillin, 1928. The most recent ones, carbapenems down here. I hope you can see that. This is not working. There we go. But what you see on the top here is when resistance has been identified in the clinic. And in most cases, it's only within a few years. So even when you produce a new antibiotic class, how much time do we really have until resistance is present? How long? It's been now, it's been 30 years since a new class has been introduced. How long do we have left until we really have effective antimicrobials for? And this is kind of why antimicrobials aren't popular for pharmaceutical companies to produce. So less than 5% of venture capital investment in pharma has gone into antimicrobial development. Well, this was between 2003 and 2013, but it shows you the point, 1.8 billion compared to a total of 38 billion. And if you look on the right, you can see how much it costs to produce an antimicrobial. By about 12 years in, you've sunk $700 million, and you're only going to make profit after 23 years. You don't make very much profit either, because antimicrobials have a particular trait. You don't actually want to use them. Once you developed a new antimicrobial, you want to protect it from resistance. You don't want to use it at all, really. Keep it for your emergency infections. But how is a pharmaceutical company meant to make any money out of that? They don't, so they stop making them. And now we have a crisis. So I've just told you how we have an absolute crisis in available antibiotics. However, worldwide, the vast majority of antibiotics are used in agriculture. And this statistic is actually from the United States in 2014, where 70% of all produced antibiotics or consumed antibiotics there were done so in agriculture. The vast majority of that is in animals. It's worth mentioning that you can use antibiotics in crop production too. For example, they were spraying orange trees with antibiotics, which is quite controversial. But anyway, that use has contributed to a very high level of resistance in bacteria that are present in the gut of food-producing animals, such as chickens, pigs, and cows. And why is this? Why do farmers use antimicrobials in agriculture? Well, there are a few genuine reasons as to why you would do this. So, for example, if your animals are sick, it's for their welfare, you want to treat them. So therapeutic treatment and metaphylaxis, which is the practice of treating animals that have been in contact with a sick one with antibiotics, is a valid use of antimicrobials in agriculture and veterinary medicine. But that often leads to prophylactic treatment, where you're just giving antimicrobials all the time to stave off the onset of a bacterial infection. This actually becomes even more insidious, because if you give animals low dosages of antibiotics, they grow faster. We don't really know why, but they do. And it's usually subclinical doses that are used. This is actually like the perfect environment for resistance to develop, because you have a very low barrier for the bacteria to get over. They just have to be a little bit resistant to survive, and from there it develops. Okay, so I'm actually looking specifically at poultry. That's what my project is on. So I'm just going to tell you about what bacterial pathogens infect poultry and why it's quite bad for farmers to have them present and not be able to treat them. And then I'll talk to you about human pathogens that come from chicken meat as well. So in chickens, you have a few major pathogens. They include Pasteurella multicida, which causes fowl cholera, Avibacterium palligallinarium, which causes infectious causa, and there's two salmonella-based diseases called pullurum disease and fowl typhoid. But one of the most important ones, the one that my research project is based around, is avian pathogenic E. coli, or APEC, which causes colibacillosis. So APEC is one of the most significant bacterial pathogens of chickens, as I said. It's responsible for about 70% of mortality in chickens when they've just been laid, so in their first week of production after they've been put into the barn. And it also causes a wide variety of syndromes. You often don't know if it's APEC or what APEC's causing it, because it can cause anything from foot pad lesions to septicemia. This variety in APEC makes it quite difficult to vaccinate against. And therefore, right now, because we have widespread antibiotic resistance and we want to discourage antibiotic use in farms, you have to kill your birds if they get infected with APEC. There isn't much else you can do. Of course, it's not just animals that suffer with resistant bacteria present in their gut. We can suffer too. If anyone's had food poisoning before, but Campylobacter is the most common cause in the UK and the Western world. It's generally self-limiting, but if it gets really bad, you need to take antibiotics to treat it. And lo and behold, Campylobacter has recently become very resistant to what was previously the #1 treatment, which is for fluoroquinolone treatment. So now we're having to change our treatment regimens because of that. You have Salmonella, which is infamous. It causes about 94 million cases per year, and there are an associated 155,000 deaths there as well. That's transmitted through raw eggs, dairy, or improperly prepared meat. And then the one I've mentioned before, I might not have done, MRSA, which is one of those famous hospital-acquired infections. Staph aureus can cause food poisoning too. And if you have resistance that develops in a bacteria or staph, which is present in chickens or pigs or whatever, there is a chance that could maybe spread over to a clinical strain. So you're just worsening the problems we already have. So this slide is a lot here, but I said that you can spread antimicrobial resistance from a staph on a farm to a staph in the clinic. How does it actually work? Well, on this diagram, each of these little pill bottles here, I'm trying to get this to work, there we go, is where you're applying antibiotics. So you have it in agriculture, in the farm, in the clinic, in companion animals, in the community, and in aquaculture. And before I keep going, I should mention that the most important source of antimicrobial resistance in the clinic is clinical use. That makes sense, really. I don't want it to sound like agriculture is dooming us all, because that isn't really true. It's just something you want to avoid, because you basically are contaminating the environment with resistant bacteria. You see, from animals, you have food products, which we then consume. That's one mechanism by which you can spread bacteria. But also, their waste is used to fertilize crops. and that can run off into waterways, or those crops themselves can be consumed. Aquaculture is a really interesting one to mention because in certain countries, huge quantities of antibiotics are used in the water to keep their fishes healthy, because when you have so many fish close together, they get sick really easily. And I'm going to come on to later why having these intensive farming practices has made the development of antimicrobial resistance that much easier. But firstly, how do bacteria actually become resistant? I'm talking about it a lot. What does that actually mean? Well, bacteria generally become resistant between one of these four mechanisms, classically anyway. You could just have a bacteria which is impermeable to an antibiotic. For example, some bacteria have two membranes, so you can't have antibiotic passing through it. Efflux pumps are proteins that pump molecules out of the cell. They often recognize molecules that have certain charges, so antibiotics are often quite similar in this regard, and they can be pumped out generally by multi-drug efflux pumps. Those are particularly nasty because once they get into a clinical isolate, you have then widespread resistance, or it makes it a lot easier for the bacteria to become resistant to other antibiotics. You can have a mutation in the target of the antibiotic. So that could be a gene, for example, which is involved in the production of proteins, or DNA, which is the target of an antibiotic. Or you can have the bacterium actually producing a gene product, a protein, that can inactivate your antibiotic. So that means it can break it down, or it can add something onto it to stop it from acting on its target site. And once you have one resistant bacterium, it doesn't take very long for that to spread. So this is a little diagram that shows it quite simply. Here we have some sensitive bacteria and a resistant one. You apply antibiotics, you're killing all the sensitive ones. Your resistant bacteria fill that niche, and then resistant bacteria can actually transfer their genes from themselves to another bacterium, sometimes a completely different species. And so if you have a lot of resistant bacteria around, you start to find those things getting swapped between different species, and that's where things start to get really nasty. The transfer of a gene from one individual to another is called horizontal gene transfer, and it's something that bacteria do quite a lot of. Antimicrobial resistance is something we study, where we study this in the context of that, because it's worrying how you can have one species transfer something to another one. It happens through three mechanisms usually. So you can have the uptake of DNA, which is released by cell lysis. For example, Campylobacter does that. So you're just absorbing the DNA from the environment. There might be a resistance gene there. Viruses, so bacteriophages or viruses of bacteria, they can carry DNA with them. So they can sometimes carry resistance genes too. But one of the most important ways in which resistance is spread is through something called a plasmid. Plasmids are extra chromosomal. That means they're a bit of DNA in the cell that isn't part of the bacterial chromosome itself, that is independently replicating and can actually spread between bacteria through conjugation, and they carry lots of resistances with them often. So we have a situation where you can have resistance in one species that develops. It might be a perfectly benign commensal, something that lives in your gut, something that's even beneficial, but something nasty comes along, a pathogen, and it just so might inherit one of these resistance genes. So this is why it's important to monitor how much resistance this level is in the population. And intensive farming has, you could say, increased the spread and the dissemination of resistance genes. So this diagram here is quite a cool one, I think. Basically, what it's trying to show you is that when you reduce the diversity of hosts, so if you keep a lot of animals in the same species together, or a lot of animals that are very genetically similar, you're allowing pathogens, and that includes bacteria, to spread much more easily through the population, and therefore resistance spreads a lot more easily through the population. And a lot of this comes from the way that we design farms. So if you're using antibiotics regularly, if waste isn't disposed properly, so they're living in their waste for periods of time where bacteria will be present, if they're housed very close together. So these things can accelerate the spread and development of resistance in bacteria. So there's a lesson to be learned here. I've got an example just after this slide. I keep getting ignored here. This is something I wanted to show you actually before I keep going about. I've talked a lot about different elements. It get a bit confusing about genes, about plasmids, about bacteria, about species, but basically it's a hierarchy. You have the little Russian dolls, which are showing off how this hierarchy works. At the very bottom, we have the gene. This could be something which produces an enzyme that breaks down an antibiotic. These can then get absorbed into bigger elements. So I won't go to teach about integrons, but they take multiple resistance genes together and democratize them and make them cheaper, for the cell to maintain. You have plasmids, which can transfer those between different bacteria, but also maintain them as well. Then you have your individual clones. For example, E. coli sequence type 131 is spread throughout the entire world. Certain pandemic clones of bacteria are resistant to a lot of different things. But they're also a part of a gene exchange community. So your resistance is being transferred between different species. That happens within a person or an animal, like myself. If I'm unfortunate enough to go to a hospital, then I am then part of that hospital's resistance profile. I have all these different bacteria there. You have, and that's how you get these resistant pathogens coming through. You have lots of antibiotics being used in that environment. So you're naturally going to have bacteria that pervade and cause disease as well. Okay, here's my my example is coming soon, I promise. I wanted to stress before I get there about how AMR, so if you can't see the titles here, I don't know if this is blocking, I'm not sure I can get rid of that. I apologize for that, I just noticed. But what that says is that AMR is a global health problem, okay? So before I talked about farms, I talked about agriculture. We can think about AMR in two contexts. In one, it's a One Health issue. That means One Health is basically the study that the health of our environment, the health of our animals, and the health of humans is all intertied together. So, if we have lots of resistance developing in farms, that might spread over to the community, someone goes to the hospital, that gets spread to the hospital, etc. But it's also a global problem because of how mobile our world is, well, until maybe the last couple of years with COVID. But anyway, you have travel, trade, sometimes even people have suggested that birds might bring bacteria with them between continents. So if you have resistance developing in one country, it's not just a problem for that country, it's a problem for the entire world. And I'm going to show you an example now. I'm sure this next slide about how that is the case. Here we go. Okay, so this example is about a resistance gene. The resistance gene is called mobilized colistin resistance 1, MCR1. And colistin is an antibiotic, which is very important because it's one of the last lines of defense against extremely resistant pathogens, gram-negative pathogens such as E. coli. And in China, in 2011, they described the first resistance gene that defended against colistin that could be transferred between different hosts. And it was described around this area originally. It's an area where they do a lot of intensive pig and chicken production. And at the same time, they found it in the diarrhea of patients. They also found it in retail meat and on farms. So you can't say for sure where it came from, because again, as I said, this is an environmental issue. AMR is being spread all over the place all the time. But there's a lot of evidence that says it could have started in the farm, because in China, they use colistin to grow chickens, they use it to grow pigs, even though it's a last line antibiotic. And it's one of the last lines of defense against really nasty infections. That said, I'm not trying to put the blame entirely on China, because in 2010, if you look at this map, this is showing you the usage of antibiotics by different countries. So Europe looks pretty suspect there, doesn't it? It's very red. North America too. We have been using a lot of antibiotics as well. Of course, the class matters, what type matters. If you're using like some basic first generation cephalosporin, something like penicillin, ampicillin, there's a lot of resistance to that anyway, so it's not as a worry if their resistance is developing. But if you're using something like colistin, or one of the really important antibiotics, things get nasty. And so what are we actually going to do about this? Because this sounds quite bad at this point, doesn't it? Well, in the last few years, so 2010 was when that data was from, in the last few years, we've been able to cut down the amount of antimicrobials we're using in UK farming and across Europe as well. So this graph shows you the amount of antibiotics that are used. 2/3 of them are used in people, a quarter are used in farm animals, a certain amount was used in either companion or farm animals. But you can see that they've decreased that amount a lot. In poultry in particular, they decreased antibiotic use by 82%, which might make you ask, did we need to use that much in the first place? We really didn't, because they were also able to increase the production by 10% as well. But it's not quite so simple as that, because even though they've reduced the amount of antibiotics that are being used through stewardship programs, what we're actually going to see is that resistance is still present. It's not really going away as quickly as we thought it would. So these little graphs here are from the EU, because they have good data on this sort of thing. There's some UK data as well. But basically, they show you 4 different types of resistances in different countries. And these are bacteria that have been taken from broilers, so chickens that you rear for meat. And basically, what we can see over time is that 2010, 2012 was when pressure started getting put on. 2014 is a date that's used a lot for the introduction of these measures, you don't see really in most countries a big change. In some countries, it looks pretty good. For example, the Netherlands here, it looks pretty good until recently. But has it really gone down very much? In some countries like Spain, you have really high resistances to certain types of antibiotics, and it's not changing. So we need to do something about this. We need an active intervention which can help us remove resistance. Oh yeah, this is something I did as well. I looked at the data myself and I looked at resistances in Europe based on the genomes of different bacteria. And what you can see is that over like 4 years, so you have 2014-17 here, there's data available for those years. There's not really, I'm trying to get the laser pointer up, but there's not really a significant change in any of the classes between those 2 years. In fact, it almost increased in that period. So it's not a good sign. And there's also something else to remember too. This is a very wordy slide, actually, I apologize for this now, but I have to really say it. And it's not my fault because the WHO has made this a horrible acronym. Some antimicrobials are classed as highest priority, critically important antimicrobials, HPCIAs. And this is really used in the context of growing, producing animals to eat. Basically, they are designated on 2 criteria. The important one is that they are possibly transmitted from non-human sources, or they have resistance genes from non-human sources. So they include quinolones, fosfomycin, and 3rd generation above cephalosporins. Those are like similar to penicillin. They're more advanced than that. But basically, what I'm looking at is this resistance in chickens. And because resistance to those is really bad, because if that spreads over to humans, these are the antibiotics we really need to look after. And if that's present and that's sticky around in bacteria, we need to do something about it. And how can CRISPR help? That's what that says. I know you can't see it. How can CRISPR help? Well, despite the fact that we've reduced the amount of antibiotics available, there's actually a problem for farms too, because whilst you don't want farmers to use antibiotics in large quantities, they still do need to use them. They need to be effective. So stewardship has made it actually more difficult to treat bacterial infections. And bacterial pathogens, like I said, cause a loss of productivity in farms, and they can pose a risk to human health too. It can then be spread throughout the environment, so you have all kinds of issues starting off there. And interventions such as CRISPR-based stuff, and we come back to CRISPR again, could help reduce the burden of AMR. So, and this all comes back to the origin of CRISPR. I'm going to go into a little bit more detail here, and this might be a bit too much, but I'll see how we go. CRISPR-Cas, I explained to you before, you have these repeats that have spaces in between them, okay, that are specific to invading elements. It's an adaptive immunity system, a bit like our immune system, but not really. It's an interesting one. Basically, what happens is an element, so up here, I'm trying to get this at the end, but there we go. A phage or a plasmid injects its DNA into the cell. That's then recognized by some CRISPR-associated genes, integrated into an array, expressed, and then it complexes with a few different things. It cuts and destroys that. But it destroys that at the point. In eukaryotes, that's repaired. In bacteria, the whole point is it isn't repaired because you're trying to kill the thing that's coming in. So if you retarget this against the host, you can actually kill them. And this is a couple of ways in which you can do that. So the double strand breaks that you're inducing are fatal if you target the chromosome. And you can just simply package your CRISPR-Cas DNA. So I said before about how you can transfer DNA between bacteria. We use this too in research to introduce things into stuff in the lab. You can use it in the field as well. This is a little bacterial phage, bacteriophage there. He has got Cas genes and CRISPR. They are being expressed in the cell and they kill the cell. Or if you have a resistance plasmid, for example, you can cure it. And then you have quite a neat use, actually, of CRISPR. This is another example, endogenous CRISPR-Cas self-targeting, where basically you don't need the Cas genes because they're already present. You remember I showed you before that slide with that huge diversity of CRISPR-Cas genes or CRISPR genes that are present. Basically, here's an example of one. I'm trying to get this to work still, but it's playing up on me, so I might have to just go without it. There you go. This is our cascade. You have multiple Cas genes present in a lot of bacteria. They can actually target the cell as well. So you can kill the cell with its own machinery, which is pretty cool. You can also introduce them from another bacteria. And we could repurpose this against plasmids. Those resistant plasmids I was telling you about before are one of our biggest problems when it comes to AMR because they accrue lots of different elements, lots of different resistances. and they then transfer themselves. This is a nice little picture of how it works. Here's one bacteria, is expressing a pilus, and that's going to this bacteria. The DNA is transferred through that, and then the plasmid can replicate in the other one. If you had a CRISPR system here, the DNA would get spread across, but it wouldn't be able to replicate because it would get broken down. So you can reduce the ability of plasmids to spread between the population and actually remove the ones that are resistant as well by targeting these elements. This is an example of what I'm trying to kill. This is the bacterial plasmid I'm interested in right now. They're pretty complicated elements. There's about 100 genes there, these arrows are genes. You have a number of genes involved in replication of the plasmid, but the ones in red are resistance genes, and these orange ones are associated resistance too. You can see they can carry lots of them, and they basically, once they're present in a bacteria, stay there because they produce genes which will kill the cell if they're lost. It's quite cool. You can talk about that for ages just by itself. But I just wanted to show you what these things look like because I'm talking about them a lot and it can be hard to visualize it. I don't know if this helps, but they're just big circular elements of DNA that contain lots of different genes in them. And we want to kill those using CRISPR. So how would you actually do that? Well, you might have heard of probiotics, which are bacteria that you eat to improve your gut health. You can actually put a plasmid onto one of those. This is quite cool. And we're talking about plasmids in a lot of different ways here. But if you put it on a probiotic, it can then transfer itself across the different bacteria. So in this diagram, I'm trying to show you the red ones as being resistant, the green ones as being sensitive. You have your little plasmids that have CRISPR on them. They go across, and if it's present, if there's resistance there, they kill it. If it's not, they don't. They can also keep spreading, and they'll eventually wipe out the resistant bacteria. So in theory, what you're actually doing, if you remember back to that slide where I showed you about how resistance develops in the first place, you're vacating a niche which then resistant bacteria will fill. In this case, we're doing the opposite. We're putting pressure on those bacteria, and your sensitive ones are going to fill the gaps. I am a bit biased. Is the best alternative to antibiotics. These are some other ones. I've always wanted to show these because these pictures are so beautiful, really. I've talked to you a bit about bacteriophages. Viruses are bacteria. Here they are here, like little space landers or something. They can infect cells and replicate and kill them in their own right. There's also another really cool little thing that people are researching. In fact, DARPA are researching this. This is a Bacterial vibrio bacteriovirus, and that is a parasitic bacteria that eats other bacteria. And so if you can target that against pathogens, you know, then we can use bacteria against bacteria. You can say that using CRISPR is bacteria against bacteria because it comes from them. But anyway, that is pretty much what I have to say today. So if I had to go back over some of the things that I've talked about, I think one of the important takeaways today is that AMR is really a big challenge that's coming up. And we're going to need different solutions, some interesting thinking to try and get past it. It's also an environmental issue. You know, this is something which isn't just present in the clinic or in farms, it's actually everywhere. Every time you're using antibiotics, you're increasing pressure, you're introducing genes to the environment. And we have monitoring programs, which are great. We have plasmids that are persistent, which we can't get rid of. So CRISPR-Cas, this great technology, which I talked about earlier, could maybe help us. That's what I'm trying to do. And for me, for my research, what actually really is there to do? Well, I talked about how we deliver it using a probiotic or some such. That's actually quite difficult. Getting that into the gut of a chicken past the stomach acid, for example, is quite hard, you know? So we have to think about delivery. Also, some bacteria have stuff in them which stops CRISPR from working. So you have to consider this as well. Are those going to complicate the situation? But yeah, that's basically what I've had to say today. Before I finish, I'd like to say thank you to all of my supervisors. Here they are here. Simon's here tonight with us. But we have lots of people here. We have Dr. Caroline Reimer from Reading, and Simon, of course, I said before. Dr. Arno van Vliet and Professor Roberto Laragioni from Surrey. And then Folium Science, who are the case partner for this project. We have Dr. Holger Newper and Professor Martin Woodward. Thank you very much. And I'd love to take your questions. Yeah. Thank you.
Thank you, Joss. Thank you very, very much. Despite the technology militating against you at every step, you've given us an absolutely brilliant lecture, and you've solved a lot of unknown questions, certainly for me, about CRISPR and CRISPR-Cas9. In fact, don't be despair about the numbers of people here. There's an awful lot of people at home also watching this on Zoom. So that would have been a full lecture for this tonight had it not been for the circumstances. Right. Can I take some questions from the floor? Would anybody like to start? Barbara.
It's not so much a question, just more a comment on whether they really work or not. And your point about how difficult it is to get microbes in an already established gut microbiome. But the data is showing that in most cases, a small amount will go in.
Yeah, so that's absolutely true. What you see is that if you introduce a bunch of bacteria, like a probiotic, most of them just go straight through your system, right? But the idea is what will happen is even if they don't stay themselves, they're passing on that plasmid to your native bacteria, which are already inhabiting that niche. And there are obviously issues of efficiency. How many plasmids are getting transferred? Is it enough? This is what we need to actually look at. But you can do things like basically, if you let the plasmids keep going, which is a problem in itself, because if you let these things run wild, you don't know where they're going to end up. They could go from one bacteria to another. You don't need that many to actually get there in the first place, to start going through and wiping out your resistant bacteria. But yeah, it's funny you mentioned that company because, you know, lots of our understanding of CRISPR actually came from yogurt research because you have tons of phages that are a big problem in yogurt research because you need bacteria to make yogurt. Phages will kill all the bacteria. And so one of the first, from my memory, one of the first papers that was looking at how we can stop phages killing our yogurt bacteria was looking at CRISPR as well. So it's a little fun anecdote for you.
I have a question of my own. Who looks after the safety side of the technology? For example, a fairly obvious question, if one person has 3 or 4 different variants of CRISPR technology going on in their intestines, who's going to check that they are all safe together?
Oh, well, so that's a good question. So in theory, right, CRISPR systems that are being transported by these bacterial vectors, so that could be a plasmid or a virus, they should never be able to get into a mammalian cell. This can't happen because those things don't affect mammals. So what you're really asking is, are these CRISPR systems, A, are they going to persist? So right now our idea is to have them transfer once and then eradicate themselves, right? If they get to a cell where they're going to kill it, they die as well. Of course, if they keep persisting, then you have problems about GMOs in the environment, et cetera, et cetera. Or is the damage you're doing to your microbiome bad for your health? That's another question, I suppose. If your CRISPR systems are killing things they shouldn't, does that give you problems? But what I'll raise to that is that antibiotics are absolutely terrible for your microbiome. It's like dropping a bomb on your gut bacteria because they're wide spectrum. They kill lots of stuff. And in fact, a lot of syndromes in hospitals start off because you use a treatment of antibiotics. You get something like C. diff that fills that niche. And that can be recurrent. That can be months of recurrent diarrhea caused by C. diff. So that's actually something I maybe should have mentioned in this talk, is that you can think of CRISPR-Cas as being like a surgeon's knife and antibiotics being like a nuclear bomb when it comes to managing what's in the microbiome.
Yeah, you mentioned earlier wheat, but you obviously aren't dealing specifically in that. But one of the areas that's interested me for a long time is work that's being done, I think, by somebody called Professor Oldroyd, on manipulating the wheat genome, particularly to fix them with CRISPR or not.
Well, I'd say absolutely. So once you understand the pathways that go into, well, anything, basically, I talked about different examples in animals, things like increasing muscle production, could be improving nitrogen fixation. If you understand those pathways, you can manipulate them. Of course, organisms are really complicated. So sometimes, like most of these examples, is something quite simple. They knocked out one gene that did one thing that was good. And something like nitrogen or in metabolism, you often have many, many different enzymes that are involved. And sometimes changing the proportion of one, there might be a rate limiting step down the road that doesn't actually change your end state. But this tool is so versatile. Once you can get it into an organism, you can edit anything. You just need to know what to do. So the problem, you have so much power. The problem is then actually, what do you do? Where do you target? So I think CRISPR will absolutely help with a lot of plant breeding programs. So people have already been trying to use it. I didn't include it here, but they've been trying to use it to produce plants that are more drought resistant or that might, after harvest, last longer, just by changing the composition of what's in there. I think the asparagine one is quite a nice example because it was just one thing they knocked out. which made them, have less asparagine. So yeah, but it's not my field, so I'm afraid I can't say any more than that, but I can say the potential is definitely there.
Yeah. Yes, sir.
You talked at the end about alternatives, and phages was one of them. I don't know a lot about this subject, but I understand also that phages are a bit like a surgeon's knife, going, like you're suggesting.
Yeah.
So could you just explain I know you said you're biased, but the advantages and disadvantages?
Yeah, of course. I'm actually really interested by phages myself. It's what I did my undergraduate project in, so I can talk a bit about that. Phage therapy is something that's been quite a hot topic for a while because of the aforementioned crisis of antibiotics. But there are a few, fundamental problems that people have to overcome first. So one of them is the same problem with CRISPR, is getting it, if it's an infection that's in the gut, it's getting it there. And effectively, because phages themselves are broken down by gastric juices, they don't efficiently get through to the gut. That's one problem. You said that they are like a surgeon's knife. That's because they have generally a very narrow host range. So there'll be something on the bacterial cell, a surface receptor, that the phage binds to, and then it does its thing and it kills the cell, right? But it's actually quite easy for bacteria to grow resistance to phages because there's a little mutation in that receptor. And then that stops. So phage bacteria ecology is a hot topic right now, understanding the balance between those. Can use a cocktail of different phages that have slightly different specificities to kill off your target. And if you know what your bacteria is really well that's causing the infection, then you can often find, companies specialize in finding phage cocktails to treat really, really difficult to treat infections. But their best use right now, I think, is topically. So because you can't get it easily into the gut, I think you can maybe do it intravenously, but it's mostly like topical infections, the stuff that you can treat on the skin. But yeah, CRISPR and phages, I talked about delivering CRISPR through a phage as well. It has the same problems of just using a phage, they've got to actually get into your bacteria that you want them to. What we're trying to do is something basically quite interesting. I might just give me a minute to go on a little tangent here. Some plasmids that I was describing before that can conjugate between bacterial cells, they produce something called a pilus. So on your cell, you have the pilus that they produce to go somewhere else. Some phages recognize specific piluses that are related with the plasmid. So if you choose the right phage, you can target that one plasmid. That's something I want to do anyway. So in a lot of ways, the technology between phage therapy and CRISPR antimicrobials, they have similar issues and advantages. I can't say which one's better. I think phage therapy's had a lot of trials done into it now. It hasn't really got what people hoped it would. People have been using it in Georgia and Russia for like 50 years, so it can work. But I don't think any of these treatments are really going to be quite as brilliant as antibiotics were. If that answers your question.
I think it's important to point out, isn't it? We're mentioning earlier this TED talk by Bonnie Breslau.
Bonnie Breslau.
Yeah. From Yale, I think, wasn't she? With the bobtailed squid and the Vibrio fischeri. But she pointed out that it's only a few bacteria that are actually harmful. Most of them are actually doing an awful lot of good. And if you look at all the DNA in all the DNA of all the bacteria in our systems, their DNA actually constitutes more in terms of base pairs than our own DNA. So we're more bacteria than we are people, really. I think it's worth mentioning that, isn't it? That you make the bacteria out to be a sort of a whole disturbance sort of sci-fi.
I don't want to do that. I love bacteria. I study microbiology. And I've been talking about doom and gloom a lot, but you're absolutely right. Bacteria are an important part of our gut health. So the relationship between our gut and our microbes is very important. It's one of those negatives about antibiotics, like I said before as well. If you kill all those lovely microbes that are great for you, then they might not come back again. And there's even been studies that have suggested that use of antibiotics early in life could lead to obesity later on, things like that. There's even research going into how our gut microbes can affect our brain chemistry. So they produce analogs to neurotransmitters and things like that. So maybe you really are what you eat, what the bacteria are like.
Anyway. Can I ask you a question about plasmids again?
Yes.
I think you alluded to the fact that plasmids were sort of viruses, but not. Is that right? Are they just random bits of DNA of their own reminiscence? Or
they're absolutely fascinating. I think if we're talking about things if you're thinking from like a mammalian perspective, the closest thing to a plasmid would be a virus, because they spread between cells. They're often like parasitic. They don't actually give benefits to the host all the time, but sometimes they do. They give you a resistance phenotype, right? That's part of the reason they stick around. And there's actually an interesting question as to how plasmids have survived and they keep going. It's called the plasmid paradox, because these things are negative, even though they can keep themselves in the cell. Basically, they produce a toxin which would kill the cell if the plasmid was lost because they produce an antitoxin to it. It's like holding a gun to the bacteria's head. But even with these things, they shouldn't really exist. They're really interesting. And they aren't part of the chromosome. They are a bit of DNA, but they're independent. They replicate independently of the bacteria. They can transfer between the bacteria. So, you don't really think of viruses as life. You can't think of plasmids as life either, but they are independently replicating bits of DNA. So if you, that was a really roundabout way of answering your question. I'd say they're closer to viruses than anything else that you'd encounter. But they're not. But they're not. I'm not sure where the distinction came in. But viruses usually would, when they infect a cell, they lyse it. They break out and they infect more. Whereas a plasmid basically inhabits the cell and almost parasitizes it. Yeah.
And can plasmids be used, on this question, against viruses?
That's a really interesting question too. So plasmids can have many different modules on them, resistance genes, for example, metabolism genes sometimes. I think even CRISPR-Cas systems in nature are found on plasmids. I don't know about against bacteriophages, so your viruses and bacteria, but I do know examples of plasmids using CRISPR-Cas against each other, right? So in nature, this actually happens. What I'm trying to do is already being done in nature in some circumstances, but what isn't, I guess,
Well, again, like we said earlier about parsimony of nature, it really works. Use it over and over again. Yeah. Good. Well, you've got to get back to Reading. So, are there any more questions? Right. Well, thank you very much for a most stimulating and interesting lecture. And thank you for coming all this way. And sorry about the technology.
Well done for battling on.
Thank you everyone for having me. It's been a lovely experience.